Today we are going to talk about hallmark of tumor. In a normal cell, growth is under controlled rate. That is, the growth rate is controlled. Normally, if there is any damage in a cell, the repair is controlled. That is, repair is by controlled growth. But in a cancer, in a malignant cell, growth is uncontrolled. For example, this is a normal cell and here is damage. Then the repair will lead to this kind of growth, that is uncontrolled growth. For the cell to be cancerous, the proto-oncogene should be gained its function, should have gained this function. That is, there should be over-functioning of proto-oncogene, gain of function of proto-oncogene, so that proto-oncogene is converted to oncogene. That's why this leads to self-sufficiency in growth signal. That is, over-functioning or gain of function of proto-oncogene leads to conversion of proto-oncogene to oncogene, so that growth signal is self-sufficient for the growth. The second one is tumor suppressor gene. Normally, tumor suppressor gene is responsible for the suppression of tumor. However, in cancer, in tumor, there is loss of function, mutation of tumor suppressor gene. That's why there is no suppression of tumor, so that tumor cell can progress further. That is under function mutation, loss of function mutation of tumor suppressor gene. This leads to insensitivity to inhibitory growth signal. Let us consider an example of CAR for the sake of understanding the tumor. Let us say there is malignant CAR in which accelerator is applied, that is gain of function mutation of proto-oncogene, and break is failed, that is loss of function mutation of tumor suppressor gene. Malignant CAR, accelerator is applied and break is failed. Now, normally DNA damage is repaired, so that there will be no progression to cancer. But in the cancerous cell, there will be also loss of function mutation of this DNA repair gene, so that there will be no repair of gene, and this can lead to cancer. That is DNA repair gene dysfunctional. In normal cell, if DNA damage occur, then there will be DNA repair by DNA repair gene. But in cancerous cell, if there is DNA damage, there will be no repair of DNA, because DNA repair gene has undergone loss of function mutation. Let us take an example of malignant CAR. We have previously said malignant CAR in which accelerator is applied, that is gain of function mutation of proto-oncogene, break is failed, that is loss of function mutation of tumor suppressor gene, and toolbox is inhibited, that is loss of function mutation of DNA repair gene. Now, in a normal, for example, if the CAR was normal, if accelerator is applied and brake is failed and toolbox is dysfunctional, then there should be blasting of the CAR for not causing any other kind of harm. But as the CAR is malignant, whether there is accelerator is applied, brake failed, toolbox dysfunctional, there will be no killing or destroying or blasting of the CAR. Survival of cell depends on two kind of gene, anti-apoptosis gene and pro-apoptosis gene. Anti-apoptosis gene are responsible for inhibiting apoptosis, but pro-apoptosis gene are responsible for causing apoptosis. In cancer, there will be gain of function mutation of anti-apoptosis gene, but loss of function mutation of pro-apoptosis gene. That's why apoptosis is inhibited in cancer. This leads to loss of inhibition of apoptosis. Okay. Now, in malignant car, brake is failed. Accelerator applied, brake failed, toolbox dysfunctional, not being killed, that is not blasting, and multiplying in normal. Normally, look here, in a normal cell, telomere is long. With every division, telomere gets shortened so that it cannot divide forever. After shortened division, it will be unable to divide. The cell will be normal cell will be unable to divide. But look here, in a cancer cell, telomerase producing enzyme will have gain of function mutation, no telomere shortening, and this leads to limitless replicative potential. That is, it can divide for forever. There will be no inhibition of division. There will be no stoppage of division, so that it can divide infinitely, so that they are multiplying in number. Now, let us summarize. Proto-oncogene, gain of function mutation; tumor suppressor gene, loss of function mutation; DNA repair gene, loss of function mutation; anti-apoptosis gene, gain of function mutation; pro-apoptosis gene, loss of function mutation; and telomerase producing gene, gain of function mutation. Also, there are angiogenesis gene, which are responsible for formation of new blood vessel. There will be also gain of function mutation of angiogenesis gene, which leads to formation of many new blood vessels, so that there will be sustained angiogenesis. This can be remembered as all roads going to the malignant car. Normally, let us summarize the hallmark of cancer. Self-sufficiency in growth signal, which is caused by gain of function mutation of proto-oncogene, that is accelerator applied. Okay, insensitivity to inhibitory growth signal, that is loss of function mutation of tumor suppressor gene, which is which can we remember by we can remember as brake fail in the car. Loss of DNA repair gene, toolbox is lost, that is toolbox damage. Evasion of apoptosis, there will no apoptosis, that is the car is not blasting, not being killed. Limitless replicative potential. Telomerase gene will have gain of function mutation, that is the malignant car is multiplying in number. Now there will be sustained angiogenesis, that is all roads will go towards the malignant car. These are the cardinal features of neoplasm, but not malignant neoplasm. The specific feature for malignancy are invasion and metastasis. What is invasion? Infiltration of cell into the surrounding tissue is called invasion. For example, let us say this is the cell and this is the neoplasm. But in invasion, there will be infiltration into the surrounding tissue inside. Let us consider this is the basement membrane, and in neoplasia, in normal neoplasm, there is no infiltration into the surrounding tissue. But here, in malignant cell, there is infiltration into the surrounding tissue beyond the basement membrane. Now, this is the invasion. In metastasis, there will be spread of tumor to distant site, which is distant from the primary tumor. Let us say this is the primary tumor, and this is the distant site, which are not in connection. But the tumor cell from primary tumor are spread to the distant site. Now, initially there was tumor only here. Now, tumor has spread to this site. This is called metastasis. Also, the tumor will escape from immunity.